Right now, as you listen to these words, your cells are carrying genetic instructions that were impossible to rewrite just 15 years ago. Somewhere in a laboratory, an artificial intelligence is designing a drug molecule that will save someone's life in 2030. And in a hospital across the world, a patient with sickle cell disease is being cured, not treated, cured at the genetic level. These aren't headlines from tomorrow. This is today, and most of us barely noticed when it happened. Before we start, a quick roll call. Write in the comments which part of this planet you're watching from, and then just listen and enjoy. Here's the question we're exploring together. What happens when humanity gains not just years, but decades of healthy life? Not life in hospitals or nursing homes, but active, engaged, meaningful life. What happens to careers, relationships, dreams, and the very structure of society when 90 becomes the new 60? This isn't science fiction anymore. The biotechnology revolution reshaping human health is already underway, quiet and systematic, building the foundations of a transformation that will fully unfold by 2050. By the end of this series, you'll understand exactly how this revolution works, what's realistic and what's hype, who will benefit first, and how these technologies will eventually reach everyone, and most importantly, what it means for you, your children, and the world we're building together. Let's start with something remarkable. In December 2020, a 91-year-old woman in the United Kingdom received the first authorized COVID-19 vaccine. Within 12 months, billions of people worldwide had received mRNA vaccines, a technology that took 30 years to develop, but felt like it appeared overnight. Most people focused on the pandemic response, but something deeper happened. We proved that biotechnology could move from laboratory concept to global deployment in record time. We demonstrated that humanity could reprogram cellular machinery to fight disease. And we showed that radical innovation in medicine wasn't decades away. It was here, now, ready to be applied to the next challenge and the next. mRNA technology didn't start with COVID-19. Catalin Carrico spent decades working on messenger RNA, often in obscurity, facing skepticism and funding difficulties. The principle was elegant. Instead of introducing a weakened virus to teach the immune system, why not give cells the instructions to produce a harmless piece of the virus themselves? Your own cells become the vaccine factory. When the pandemic arrived, this decades-long foundation allowed for unprecedented speed. BioNTech and Moderna designed their vaccine candidates within days of receiving the viral sequence. Days, not years. This is the pattern of biotechnology revolutions. Long periods of invisible progress, then sudden, visible transformation. Right now, we're in one of those invisible progress periods for multiple technologies that will seem to appear suddenly in the coming years. Consider CRISPR, the gene editing tool that won the Nobel Prize in Chemistry in 2020. In 2012, Jennifer Doudna and Emmanuel Charpentier published their landmark paper showing how bacterial immune systems could be adapted to edit any DNA sequence with precision. By 2023, the first CRISPR therapy was approved for treating sickle cell disease. Patients who lived with chronic pain and regular blood transfusions now have their own edited cells producing healthy hemoglobin. This isn't managing a disease, it's correcting the genetic error that causes it. The technology works like this. CRISPR acts as molecular scissors guided by RNA to find and cut specific DNA sequences. Scientists remove some of the patient's blood stem cells, edit the problematic gene in the laboratory, then return the corrected cells to the patient's body. These edited cells multiply and produce healthy blood cells. One treatment, permanent results. The cost today is substantial, around $2 to $3 million per patient. But remember the pattern. The first human genome sequencing cost $3 billion and took 13 years. Today, you can sequence your genome for less than $1,000 in a few days. Gene editing is expanding beyond rare blood disorders. Clinical trials are underway for inherited blindness, muscular dystrophy, certain cancers, and cardiovascular diseases. Each success builds knowledge, refines techniques, and reduces costs. The infrastructure being created now, the trained specialists, the manufacturing facilities, the regulatory frameworks, will enable the next wave of genetic medicines. Then there's artificial intelligence, the invisible partner accelerating every aspect of biotechnology. AlphaFold, developed by DeepMind, solved a 50-year-old problem in biology, predicting how proteins fold into three-dimensional shapes. Proteins are the workhorses of life, and their shape determines their function. Understanding protein structure is essential for drug design, but determining these structures experimentally took months or years of painstaking work. AlphaFold can predict protein structures in hours with remarkable accuracy. This tool is now freely available to researchers worldwide, accelerating drug discovery across thousands of laboratories. AI systems are also designing entirely new molecules. Traditional drug development involves synthesizing and testing thousands of compounds, hoping to find one that works. Now, machine learning algorithms can predict which molecular structures will bind to disease targets before anything is synthesized in a laboratory. Companies like In Silico Medicine and Recursion Pharmaceuticals are using AI to identify drug candidates in months instead of years. The first AI-designed drugs are entering clinical trials now, in 2025. By 2030, AI-assisted drug discovery will be standard practice. What makes this moment different from previous waves of medical progress? Three factors converging simultaneously. First, we can now read biological information with unprecedented detail and speed. Genomic sequencing, proteomics, metabolomics. We can measure what's happening inside cells and bodies with molecular precision. Second, we can write and edit biological information. Gene editing, synthetic biology, mRNA technology. We're moving from observation to intervention at the fundamental level. Third, we can process and learn from biological information using artificial intelligence, finding patterns and solutions that human researchers might never see. These three capabilities, reading, writing, and computing biology, are reinforcing each other. Better AI helps us understand complex genomic data. Better understanding enables more precise editing. More precise editing generates new data to train better AI. This feedback loop is accelerating progress in ways that aren't always visible from the outside. Why is 2050 the realistic horizon for transformation? Because biotechnology doesn't revolutionize health overnight. It moves through predictable stages. First comes basic research, understanding biological mechanisms. Then proof of concept in laboratory settings. Then small clinical trials testing safety. Then larger trials proving effectiveness. Then regulatory approval. Then manufacturing scale-up. Then gradual cost reduction. Then widespread adoption. This process takes time, typically 15 to 20 years from initial discovery to broad availability. The technologies entering clinical trials today in 2025 will reach widespread use in the late 2030s and 2040s. The discoveries happening right now in research laboratories will become the standard treatments of the 2040s and early 2050s. This timeline isn't speculation. It's the established pattern of how medical innovations diffuse through society. Look at statins, the cholesterol-lowering drugs. First discovered in the 1970s, they didn't become widely prescribed until the 1990s and 2000s. Now, they're routine preventive medicine, extending millions of lives. Or consider in vitro fertilization. The first IVF baby was born in 1978, amid enormous controversy and ethical debate. Today, IVF is standard reproductive medicine, with over 10 million babies born through assisted reproduction. The controversial becomes routine, the expensive becomes affordable, and the rare becomes common. This is how medical revolutions unfold. We're standing at a similar threshold now, but for interventions far more fundamental than lowering cholesterol or assisting conception. We're developing the ability to rewrite our genetic code, regenerate damaged tissues, engineer our microbiome, and extend our healthy lifespan. These aren't distant possibilities. They're emerging realities with clear development pathways. But here's what matters most. This revolution is quiet because it's built on decades of patient scientific work. There are no miracle breakthroughs, only the accumulation of knowledge and technique. Every gene editing success builds on thousands of experiments that failed or succeeded partially. Every AI breakthrough in drug design relies on massive datasets painstakingly collected over years. Every mRNA vaccine stands on decades of research into lipid nanoparticles and immune responses. The biotechnology revolution is already underway because the foundations were laid long ago, often by researchers working in obscurity, driven by curiosity and persistence rather than headlines or hype. What we're experiencing now is the moment when all those separate advances converge, when the infrastructure is finally in place, when the tools are finally powerful enough, and when society is finally ready to embrace these changes. In the episodes ahead, we'll explore each dimension of this transformation in detail. How gene editing will evolve from treating rare diseases to preventing common ones. How longevity science is shifting, aging from inevitable decline to treatable biology. How personalized medicine will replace one-size-fits-all treatments. How regenerative medicine will make organ failure a solvable problem. How our microbiome will become a new frontier for health interventions. And crucially, how these technologies will spread from wealthy early adopters to become globally accessible. We'll also face the harder questions. Who benefits first and why? How do we navigate the ethics of human enhancement? What does it mean to live not just longer, but differently? These aren't questions with simple answers, but they're questions we need to explore together as this revolution unfolds. The future of human health isn't arriving all at once in some dramatic moment. It's arriving gradually, systematically, built on the quiet work of thousands of researchers, clinicians, and innovators around the world. Some of that future is already here, in the mRNA vaccines protecting billions, in the gene therapies curing previously incurable diseases, in the AI systems designing tomorrow's medicines. The rest is coming, step by patient step, toward that horizon in 2050, when we'll look back and realize that everything changed, even though we barely noticed it happening. There's a moment in every revolution when the impossible becomes routine. For gene editing, that moment arrived quietly in 2023, when regulatory agencies in the United Kingdom and United States approved Casgevy, the first CRISPR-based therapy for human use. Patients with sickle cell disease and beta thalassemia, conditions that had meant a lifetime of pain and medical intervention, could now be cured with a single treatment. Not managed, cured. The genetic error that had been passed down through generations could simply be corrected. Let's understand what actually happened, because the reality is more fascinating than any simplified version. Sickle cell disease occurs when a single letter in the genetic code is wrong. Just one nucleotide out of 3 billion. This tiny error causes red blood cells to twist into sickle shapes that block blood vessels, causing excruciating pain, organ damage, and shortened lifespan. For decades, treatment meant managing symptoms, pain medication, blood transfusions, hydroxyurea to reduce sickling episodes. Patients lived with their disease. Now, we can rewrite that one wrong letter. The approved therapy doesn't actually fix the sickle cell mutation directly. Instead, it uses CRISPR to reactivate a gene that normally shuts off after birth. Humans produce fetal hemoglobin in the womb, which works perfectly well in sickle cell patients. But shortly after birth, our bodies switch to adult hemoglobin, and in people with sickle cell disease, that adult hemoglobin is malformed. The CRISPR therapy turns the fetal hemoglobin gene back on permanently. The body produces healthy fetal hemoglobin throughout life, and the sickle cell mutation becomes irrelevant. Here's how the treatment works in practice. Patients first undergo testing to confirm they're suitable candidates. Then doctors collect their blood stem cells through a process similar to blood donation, but more intensive. These stem cells travel to a specialized laboratory where technicians use CRISPR to edit the specific gene controlling hemoglobin production. The edited cells are grown and multiplied in culture, creating millions of corrected stem cells. Meanwhile, the patient undergoes chemotherapy to clear out their existing bone marrow, making space for the edited cells. Finally, the corrected stem cells are infused back into the patient's body, where they establish themselves in the bone marrow and begin producing healthy blood cells. The entire process takes several months and requires hospitalization. It's not simple, but the results have been remarkable. Patients in clinical trials have remained free of pain crises for years after treatment. Their bodies are producing healthy red blood cells continuously. They're living without the disease that defined their entire existence. This is just the beginning. CRISPR technology emerged from a surprising place: bacterial immune systems. Bacteria face constant viral attacks and developed a sophisticated defense mechanism. They capture pieces of viral DNA and store them in their own genome as a kind of genetic memory. When that virus attacks again, the bacteria use this stored sequence as a guide to find and cut the viral DNA with precision. Scientists Jennifer Doudna and Emmanuel Charpentier realized this bacterial system could be adapted into a universal gene editing tool. They published their landmark paper in 2012, showing how to program CRISPR to cut any DNA sequence. What made CRISPR revolutionary wasn't that it was the first gene editing tool. Earlier technologies like zinc finger nucleases and talons already existed. CRISPR was revolutionary because it was simple, cheap, and remarkably precise. A laboratory that couldn't afford expensive gene editing equipment could suddenly edit genes with basic molecular biology tools. Research accelerated globally, almost overnight. Within 2 years, labs worldwide were using CRISPR. Within 5 years, it was being tested in human cells. Within a decade, it was curing patients. But CRISPR isn't perfect, and understanding its limitations matters as much as celebrating its successes. The primary concern has always been off-target effects. The CRISPR system might occasionally cut DNA at unintended locations that look similar to the target sequence. Early versions had measurable off-target activity. Years of refinement have dramatically improved specificity. Modern CRISPR variants, like base editors and prime editors, can make even more precise changes, altering single letters of DNA without cutting both strands, reducing the risk of unintended edits. The second challenge is delivery. Getting CRISPR into the right cells in the body is straightforward for blood diseases because we can remove cells, edit them in the laboratory, and return them. But what about genetic diseases affecting the liver, muscles, brain, or other organs? Scientists can't remove those organs, edit them, and put them back. They need to deliver CRISPR directly to cells inside the body. This is where viral vectors come in. Viruses are nature's delivery specialists, evolved to infiltrate cells and insert genetic material. Scientists have repurposed certain viruses, particularly adeno-associated viruses, removing their disease-causing components and loading them with CRISPR machinery. These modified viruses can be injected into the bloodstream or directly into affected tissues, where they deliver CRISPR to the target cells. This approach is already being tested for inherited blindness caused by mutations in the eye, for Duchenne muscular dystrophy, and for certain liver diseases. In 2023, researchers at the University of Pennsylvania treated patients with a rare form of inherited blindness called Leber congenital amaurosis using direct injection of CRISPR into the eye. Early results showed some visual improvement in several patients. The treatment didn't restore perfect vision, but it demonstrated that in-body gene editing could work. Each success teaches researchers how to improve delivery, increase efficiency, and enhance safety. Beyond CRISPR, other gene editing technologies are emerging. Base editors, developed by David Liu's laboratory at Harvard, can change one DNA letter to another without cutting the double helix. Prime editors go further, allowing insertions, deletions, and all possible base changes at a target site. These refined tools expand what's possible. Some genetic diseases require deleting entire sections of DNA or inserting new sequences. Some need extreme precision at sites where CRISPR's double-strand breaks might be too risky. Having multiple editing tools means matching the right technology to each specific disease. The pipeline of diseases being targeted is expanding rapidly. Sickle cell disease and beta thalassemia were first because they're caused by well-understood single-gene mutations in easily accessible blood cells. But clinical trials are now underway or in planning for dozens of genetic conditions. Familial hypercholesterolemia, which causes extremely high cholesterol and early heart disease. Transthyretin amyloidosis, where misfolded proteins damage the heart and nerves. Various forms of inherited blindness. Certain types of muscular dystrophy. Some rare metabolic disorders. Even some cancers, where editing immune cells can help them recognize and destroy tumors. Each disease presents unique challenges. Some require permanent editing in long-lived cells. Others might benefit from temporary editing. Some need nearly complete correction to achieve benefit. Others show improvement with partial correction. Some affect easily accessible tissues. Others hide in protected organs like the brain. The learning curve is steep, but it's being climbed methodically, one disease at a time. Now, we approach the boundary between therapy and enhancement, and this is where the conversation becomes more complex. Editing genes to cure sickle cell disease is universally accepted as ethical medicine. But what about editing genes to reduce the risk of Alzheimer's disease in someone who hasn't developed it yet? What about editing to improve muscle mass or enhance cognitive function? Where exactly is the line between treating disease and enhancing human capabilities? These aren't abstract philosophical questions anymore. The technology exists. The APOE4 gene variant significantly increases Alzheimer's risk. The PCSK9 gene affects cholesterol levels and heart disease risk. The myostatin gene regulates muscle growth. We know the genetic variants associated with height, intelligence, athletic ability, and dozens of other traits. Technically, many of these could be edited. But should they be? Most scientists and ethicists draw a clear line. Editing to treat or prevent serious disease is medicine. Editing for enhancement is something else entirely, something that requires much deeper societal discussion before proceeding. The international consensus, formalized in various guidelines and regulations, is that gene editing should focus on therapeutic applications. Enhancement editing, especially germline editing that would affect future generations, remains firmly in the realm of prohibited research in most countries. That consensus was tested in 2018 when Chinese scientist He Jiankui announced he had created the first gene-edited babies, twin girls whose CCR5 gene he modified in an attempt to make them resistant to HIV. The scientific community responded with widespread condemnation. The editing was medically unnecessary. The girls faced no immediate HIV threat. The long-term effects were unknown. The consent process was questionable. And most fundamentally, he had crossed the ethical line against germline editing without proper oversight or justification. He was later sentenced to prison in China, and the incident strengthened international resolve to maintain clear boundaries around gene editing applications. But here's the nuance that matters. As gene editing becomes more routine for clear medical conditions, the boundaries will be tested. If we can edit out the APOE4 variant to prevent Alzheimer's, is that therapy or enhancement? If we edit a gene that lowers heart disease risk by 30%, is that treating disease or improving normal health? These questions don't have simple answers, and different societies may answer them differently. What's clear is that gene editing is transitioning from experimental procedure to established medicine. The infrastructure is being built. Specialized laboratories, trained genetic counselors, clear regulatory pathways, manufacturing facilities for edited cell products, insurance coverage frameworks. In 2025, only a handful of centers worldwide can perform approved gene editing therapies. By 2035, major medical centers in developed countries will likely have gene editing capabilities for multiple conditions. By 2045, gene editing will be part of standard medical practice for dozens of genetic diseases. The cost trajectory tells this story clearly. Current CRISPR therapies cost $2 to $3 million per patient, similar to earlier gene therapies. This price reflects the complexity, the specialized facilities, the individualized manufacturing, and the recovery of development costs. But the price is already declining. Competing therapies create market pressure. Manufacturing processes improve and scale up. Regulatory pathways become more efficient once the first approvals establish precedent. Insurance companies and health systems negotiate. The same pattern that brought genome sequencing from billions to hundreds of dollars will bring gene editing from millions to hundreds of thousands, then eventually to tens of thousands. Looking toward 2050, gene editing will have expanded in three major directions. First, the range of treatable diseases will grow from rare genetic conditions to include common diseases with genetic components. Certain forms of heart disease, some cancers, various neurodegenerative conditions. Second, editing will move from ex vivo treatments, where cells are edited outside the body, to more in vivo treatments delivered directly to organs and tissues. Third, preventive editing will begin cautiously, targeting clear disease risks in people with strong genetic predispositions before symptoms appear. But gene editing won't cure everything. Most common diseases involve multiple genes interacting with environmental factors in complex ways. Type 2 diabetes, most cancers, depression, autoimmune diseases, these don't have simple genetic fixes. Gene editing will be one tool among many, powerful for certain specific conditions, but not a universal solution to human health challenges. What makes this moment extraordinary isn't that we've solved all genetic disease. It's that we've crossed the threshold from reading our genetic code to rewriting it. For 4 billion years, life evolved through random mutation and natural selection. For the first time, one species can deliberately edit its own genetic instructions. This capability, used carefully and ethically, will prevent immense suffering. It will cure children born with devastating genetic conditions. It will spare families the anguish of inherited diseases. And it will open new questions about what it means to be human when our biology is no longer entirely determined by chance. The gene editing revolution is happening now, patient by patient, disease by disease, building toward a future where genetic disease becomes increasingly rare and increasingly treatable. Here's a thought experiment that reveals how profoundly our thinking is shifting. A century ago, if someone told a doctor they wanted treatment for aging, the doctor would have looked at them with confusion. Aging wasn't a condition to treat. It was simply life unfolding. Today, in 2025, there are laboratories across the world where aging is studied not as an inevitable decline, but as a biological process with specific mechanisms that can be measured, understood, and potentially modified. This shift from philosophical acceptance to scientific intervention represents one of the most significant changes in how humanity thinks about health and longevity. Let's be clear about what we're discussing, because the field of longevity science is filled with hype and misunderstanding. We're not talking about immortality. We're not even primarily talking about maximum lifespan, the theoretical upper limit of human survival. We're talking about health span, the number of years lived in good health, free from disability and chronic disease. The goal isn't to keep people alive longer in states of frailty and dependence. The goal is to extend the period of vigorous, healthy life, compressing the years of decline into a shorter period at the end. This distinction matters enormously. Average human lifespan has increased dramatically over the past century, from around 50 years in the 1920s to over 70 globally today, and over 80 in many developed countries. But this increase came primarily from reducing infant mortality, curing infectious diseases, and improving nutrition and sanitation. maximum lifespan hasn't changed much. The oldest verified person, Jean Calment of France, lived to 122 years. Humans seem to have a biological limit somewhere in that range, and no amount of good healthcare has pushed far beyond it. What's changing now is our understanding of why we age at all. Aging isn't just wear and tear, like a machine breaking down over time. It's a complex biological process involving specific, measurable changes at the cellular and molecular level. In 2013, Carlos López-Otín and his colleagues published a landmark paper identifying the hallmarks of aging, 9 fundamental processes that contribute to aging across species. These include genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. That's a technical list, but each item represents a specific biological mechanism that scientists can now study and potentially intervene in. Let's explore the most promising areas where intervention is already showing results. Start with cellular senescence. As cells divide throughout life, some eventually enter a state called senescence. They stop dividing, but don't die. Instead, they remain in tissues, secreting inflammatory molecules that damage surrounding healthy cells. These senescent cells accumulate with age, contributing to chronic inflammation, tissue dysfunction, and age-related diseases. They're sometimes called zombie cells, alive but no longer functioning properly and actively causing harm. The remarkable discovery is that removing senescent cells can reverse aspects of aging. In 2011, researchers at the Mayo Clinic created mice genetically engineered to eliminate senescent cells on command. When they cleared these cells from middle-aged mice, the animals lived longer and healthier, with delayed onset of age-related diseases. This led to the development of senolytic drugs, compounds that selectively kill senescent cells while leaving healthy cells intact. Several senolytic drugs are now in human clinical trials. Dasatinib and quercetin, a combination of a cancer drug and a plant compound, showed preliminary benefits in small human studies for conditions like idiopathic pulmonary fibrosis and diabetic kidney disease. Fisetin, another natural compound, is being tested for its senolytic properties. These aren't yet proven anti-aging therapies, but the early results are encouraging enough that larger, more rigorous trials are underway. The key insight here is specificity. We're not trying to slow down all cellular processes or stop cells from aging. We're targeting a specific dysfunction, the accumulation of senescent cells, with drugs designed to eliminate them. This approach stands a much better chance of working than vague interventions aimed generally at anti-aging. Then there's the question of cellular energy production and mitochondrial function. Mitochondria are the powerhouses of cells, converting nutrients into usable energy. As we age, mitochondrial function declines. Cells produce less energy and generate more reactive oxygen species, damaging molecules that contribute to aging. NAD+, nicotinamide adenine dinucleotide, is a crucial molecule involved in cellular energy production. NAD+ levels decline significantly with age, and this decline correlates with many age-related problems. Researchers discovered that boosting NAD+ levels in aged mice improved mitochondrial function, increased exercise endurance, and showed various markers of improved health. This led to human trials of NAD+ precursors like nicotinamide riboside and nicotinamide mononucleotide. These compounds can be taken orally and boost NAD+ levels in humans. Multiple companies are now selling these supplements, and rigorous clinical trials are testing whether they actually improve health span indicators in humans. Here's where scientific caution is essential. Yes, NAD+ levels decline with age. Yes, boosting NAD+ improves various parameters in laboratory animals. But we don't yet have definitive evidence that NAD+ supplementation extends healthy human lifespan or prevents age-related disease in humans. The studies are ongoing. Some preliminary results are promising, but anyone claiming these supplements are proven anti-aging therapies is running ahead of the evidence. The same measured assessment applies to rapamycin and its analogs. Rapamycin is an immunosuppressant drug used in organ transplant patients, but it has a fascinating property. It inhibits mTOR, a protein complex that regulates cell growth and metabolism. When mTOR activity is reduced, cells shift into a more maintenance-oriented state, increasing autophagy, the process by which cells clean up damaged components. Rapamycin extends lifespan in essentially every organism tested: yeast, worms, flies, and mice. Mice given rapamycin live 10 to 15% longer on average and show delayed onset of age-related diseases. This is one of the most robust longevity interventions in animal research. The question is whether it works in humans and whether the benefits outweigh the risks. Rapamycin suppresses the immune system, which is problematic for long-term use in healthy people. But researchers are exploring modified dosing schedules and rapamycin analogs that might provide the longevity benefits with fewer side effects. Small clinical trials are beginning. Some physicians are prescribing low-dose rapamycin off-label to patients interested in longevity interventions, though this remains controversial and not supported by large-scale evidence. By the 2030s, we'll likely have clearer data on whether rapamycin or related compounds can safely extend human health span. Metformin, the diabetes drug, presents another interesting case. People with type 2 diabetes who take metformin appear to have lower rates of age-related diseases like cancer and cardiovascular disease than expected. Some studies even suggest diabetics on metformin live as long as or longer than non-diabetics not taking the drug, despite having diabetes. This observation led to the hypothesis that metformin might have genuine anti-aging effects beyond its diabetes treatment. The TAME trial, targeting aging with metformin, is designed to test this hypothesis directly. It's enrolling thousands of older adults without diabetes to see whether metformin delays the onset of age-related diseases. This trial is significant not just for testing metformin, but for establishing aging itself as a legitimate target for medical intervention. If successful, it could pave the way for regulatory pathways to approve drugs specifically for extending health span, not just treating individual diseases. Beyond drugs, there's caloric restriction, the most consistently proven intervention for extending lifespan across species. Reducing calorie intake by 20 to 40% while maintaining adequate nutrition extends lifespan in yeast, worms, flies, rodents, and non-human primates. The mechanism involves shifting metabolism into a more efficient, maintenance-oriented state, similar to what rapamycin does by inhibiting mTOR. Caloric restriction is difficult for humans to maintain long-term. Most people can't sustain eating significantly less food for years or decades, but it points towards something important. The body has built-in longevity pathways that respond to nutritional status. When food is scarce, organisms shift resources toward maintenance and survival rather than growth and reproduction. Scientists are now trying to develop drugs that activate these same pathways without requiring actual caloric restriction. The NAD+ boosters, rapamycin, and metformin, are all attempts to mimic aspects of caloric restriction's benefits. Intermittent fasting and time-restricted eating are more practical approaches that some research suggests may activate similar pathways. Limiting eating to certain hours of the day or fasting for 1 or 2 days per week might provide some longevity benefits without chronic calorie restriction. The evidence in humans is still preliminary, but the interventions are safe and some people find them sustainable. Then there are the more speculative but scientifically grounded approaches. Partial cellular reprogramming involves temporarily activating the factors that convert adult cells back to stem cell-like states. In mice, this approach has reversed signs of aging and extended lifespan without causing tumors, which was a major concern. The technique essentially resets the epigenetic markers that accumulate with age, making old cells functionally younger. If this can be made safe and controllable in humans, it could be extraordinarily powerful. Xenomorphic drugs, unlike senolytics that kill senescent cells, would suppress the harmful secretions from these cells without killing them. Young blood factors, compounds found in young blood that improve function in aged tissues, are being identified and tested. Stem cell therapies to replace exhausted stem cell populations are in various stages of development. Each of these represents a specific intervention targeting a specific hallmark of aging. What's the realistic timeline for these interventions to become mainstream medicine? We need to think in phases. The first phase, happening now through the early 2030s, is establishing proof of concept in humans. Do senolytics actually improve health span markers in people? Does rapamycin safely extend healthy life? Does metformin prevent age-related diseases in non-diabetics? These questions are being answered through clinical trials currently underway. The second phase, likely in the 2030s, will be refining the interventions, finding optimal doses, understanding who benefits most, identifying biomarkers to track effectiveness, developing better drugs with fewer side effects. This phase turns experimental interventions into reliable therapies. The third phase, reaching into the 2040s and toward 2050, is widespread adoption. As interventions prove safe and effective, they'll become part of standard preventive medicine. Periodic senolytic treatments to clear zombie cells, daily medications that support cellular maintenance, biomarker monitoring to track biological age and adjust interventions, annual treatments to boost stem cell populations. This isn't science fiction, it's the logical progression of current research. By 2050, the concept of treating aging will be as accepted as treating high blood pressure is today. People in their 50s and 60s won't just manage individual diseases as they arise. They'll take medications and undergo treatments specifically designed to slow biological aging, keeping multiple organ systems functioning well simultaneously. The result won't be immortality or even dramatically extended maximum lifespan. The result will be compression of morbidity, spending more years healthy and fewer years sick. What does 90 as the new 60 actually mean? It means someone at 90 having the physical capabilities, cognitive function, and disease risk profile that a 60-year-old has today. It means working longer because remaining capable longer. It means pursuing new careers in one's 70s and 80s. It means having the energy to travel, learn, and engage fully with life into very advanced age, then declining relatively quickly at the end. rather than spending decades in frailty. This vision isn't guaranteed. Biology is complex, and interventions might not work as hoped. But the scientific foundation is solid. We understand the mechanisms of aging better than ever before. We have specific, testable interventions targeting those mechanisms, and we're systematically testing them in rigorous clinical trials. The biotechnology revolution in aging isn't about magical cures or fountain of youth promises. It's about careful, methodical science, translating our growing understanding of aging biology into medical interventions that extend the healthy portion of human life. Imagine walking into a doctor's office in 2045. Before any examination, the physician already has a remarkably detailed picture of your health. Your complete genome sequence, analyzed for thousands of disease risk variants. Your microbiome composition, showing exactly which bacterial species inhabit your gut and how they affect your metabolism. Your proteome, a snapshot of the thousands of proteins currently active in your body. Continuous data from biosensors tracking your heart rhythm, blood glucose, sleep patterns, and inflammatory markers. And perhaps most remarkably, a digital twin, a computer simulation of your unique biology that can predict how you'll respond to different treatments before you take a single pill. This isn't distant speculation. Every component of this scenario exists in some form today in 2025. What's coming by mid-century is the integration and democratization of these technologies, transforming medicine from population averages to individual precision. Let's start with the foundation, your genome. The Human Genome Project, completed in 2003, took 13 years and cost roughly $3 billion to sequence one human genome. It was an extraordinary achievement that revealed the complete genetic instructions set for building a human being. Today, in 2025, you can sequence your entire genome for under $1,000 in a few days. This 1,000-fold cost reduction in 2 decades is faster than even the famous Moore's Law that drove computer chip advancement. Millions of people have now had their genomes sequenced, either through medical testing or consumer genomics companies. This massive data set has enabled genome-wide association studies, research that links specific genetic variants to diseases, drug responses, and traits. We now know that carrying 2 copies of the APOE4 gene variant increases Alzheimer's risk substantially. We know that certain variants in the SLC01B1 gene affect how the body processes statins, explaining why some people experience severe muscle pain on these cholesterol drugs, while others tolerate them perfectly. We know that variations in CYP2C19 affect how people metabolize clopidogrel, a blood-thinning drug, making it ineffective in some patients. This knowledge is already changing medical practice. Pharmacogenomics, using genetic information to guide drug prescribing, is becoming routine for certain medications. Before prescribing some cancer drugs, oncologists now test for specific genetic variants that predict response or toxicity. Before starting certain psychiatric medications, some clinics test genes involved in drug metabolism to avoid prescribing drugs the patient can't metabolize properly. The FDA has updated labels on hundreds of medications to include pharmacogenomic information. By 2035, genome sequencing will likely be routine for newborns in developed countries, creating a baseline genetic profile that follows people throughout life. When someone develops symptoms, physicians won't just consider their current presentation. They'll integrate genetic predispositions, identifying whether this chest pain in a 40-year-old with specific cardiac risk variants needs more aggressive evaluation than the same chest pain in someone with favorable genetics. They'll prescribe medications with confidence that the patient's genetic makeup supports normal metabolism of that drug. But the genome is only the beginning. Your genome is mostly fixed from birth, with rare exceptions like mutations in cancer cells. What changes constantly is which genes are active and how they're being expressed. This is where the proteome becomes crucial. Proteins are the workhorses of biology, the molecules that actually do things in cells. DNA stores information, but proteins carry out functions. At any moment, your cells are producing thousands of different proteins in varying amounts, and this pattern changes based on health, disease, diet, stress, and countless other factors. Proteomics technology can now measure thousands of proteins simultaneously from a blood sample. Companies like SomaLogic and Olink are developing high-throughput protein measurement platforms. Early applications focus on cancer detection and monitoring. Certain protein patterns in blood can indicate cancer presence before tumors are visible on scans, catching disease at the earliest, most treatable stages. Other protein signatures indicate heart disease risk, neurodegenerative disease progression, or inflammatory conditions. Imagine the shift this represents. Today, much of medicine is reactive. Symptoms appear, we investigate, we diagnose, we treat. With continuous proteomics monitoring, medicine becomes anticipatory. Protein patterns shift months before symptoms appear, providing warning signals. The protein signature, indicating pre-diabetic metabolic changes, appears years before blood glucose rises enough for diagnosis. The protein markers of Alzheimer's pathology appear decades before memory loss. The inflammatory signatures of autoimmune disease appear before organ damage occurs. This early detection window creates opportunities for intervention when treatment is most effective. It's far easier to prevent type 2 diabetes with lifestyle changes when someone is pre-diabetic than to manage advanced diabetes with multiple complications. It's more effective to clear amyloid protein from the brain in early Alzheimer's than to try reversing years of neurodegeneration. It's simpler to calm an overactive immune system before it destroys joints or organs than to repair the damage afterward. The challenge is data integration. A genome sequence contains 3 billion data points. A proteome measurement might track 10,000 proteins. Add metabolomics, measuring thousands of small molecules involved in metabolism. Add microbiome sequencing, identifying hundreds of bacterial species and their genes. Add continuous monitoring from wearable devices, generating millions of data points about heart rate, activity, sleep, and more. The total data set for a single individual becomes enormous and impossibly complex for human physicians to interpret comprehensively. This is where artificial intelligence transforms personalized medicine from theoretical possibility to practical reality. Machine learning algorithms excel at finding patterns in massive, complex data sets. They can identify subtle signatures in multi-omic data that predict disease risk or treatment response. They can integrate genetic predisposition, current protein patterns, microbiome composition, and lifestyle data to generate risk scores more accurate than any single factor alone. Several AI systems have already demonstrated impressive medical capabilities. DeepMind's AlphaFold predicted protein structures with remarkable accuracy. Google's AI systems can detect diabetic retinopathy from retinal images as accurately as expert ophthalmologists and identify lung cancer in CT scans earlier than radiologists. IBM Watson and similar systems can analyze medical literature and patient data to suggest treatment options physicians might not consider. But the most transformative application is the digital twin concept. A digital twin is a computer simulation of a specific individual's biology, built from their genomic data, physiological measurements, and medical history. This simulation can predict how that individual will respond to different treatments, allowing physicians and patients to test interventions virtually before implementing them physically. The concept originates from aerospace and manufacturing, where engineers create digital models of complex systems like aircraft engines to predict performance and identify potential failures. Applying this to human biology is far more complex, but the fundamental principle is the same. Build a model accurate enough to make useful predictions, then use those predictions to optimize interventions. Early medical digital twins are already in use for specific applications. Some cancer treatment centers use tumor simulations to predict how specific patients' tumors will respond to different chemotherapy combinations, helping select the most effective regimen while minimizing toxicity. Researchers are building heart models that simulate electrical activity and blood flow in individual patients with arrhythmias, helping plan optimal ablation procedures. By the 2030s, digital twins will expand to model entire organ systems and whole-body physiology. Your digital twin heart won't just be a generic heart model. It will incorporate your specific genetic variants affecting cardiac function, your current physiological state, your coronary artery anatomy from imaging, and your medical history. Cardiologists could test different medications virtually, predicting which would control your blood pressure most effectively with the fewest side effects before you take anything. Your digital twin metabolism could integrate your genome, microbiome, continuous glucose monitoring data, and dietary habits to predict exactly how different foods affect your blood sugar and inflammation levels. No more generic dietary advice. Specific recommendations based on your unique biological responses, tested first in simulation. The technical challenges are substantial. Building accurate digital twins requires enormous computing power and sophisticated models of human biology. We don't yet understand biological systems comprehensively enough to model them perfectly. But the models don't need to be perfect to be useful. They need to be accurate enough to make better predictions than current approaches, and that threshold is achievable with current technology and improving rapidly. Privacy and data security become paramount in this future. Your genomic, proteomic, microbiome, and continuous health data represent the most personal information possible. This data could predict disease risks, potentially affecting insurance, employment, or relationships if misused. Strong regulations and technical safeguards are essential. Several countries have enacted genetic privacy laws. The technology for secure data storage and encrypted computation exists. But maintaining privacy while enabling personalized medicine requires constant vigilance and thoughtful policy. There's also the question of what to do with probabilistic information. If your genetic analysis reveals a 40% increased risk of developing Alzheimer's by age 75, what should you do with that information? There's no certainty, just elevated probability. Some people want to know their risks so they can plan accordingly, make lifestyle changes, or participate in prevention trials. Others prefer not to know probabilities for diseases they can't prevent. Personalized medicine must respect both preferences, providing information to those who want it while not forcing unwanted knowledge on those who don't. The equity dimensions are also critical. If genome sequencing, proteomics monitoring, AI analysis, and digital twin simulations are available only to wealthy individuals in advanced economies, personalized medicine could widen health disparities rather than narrow them. The cost trajectories are encouraging. Sequencing costs have plummeted, but ensuring equitable access requires intentional effort from governments, healthcare systems, and technology developers. Looking toward 2050, personalized medicine will likely operate at multiple tiers. Basic personalization, adjusting medication doses based on genetic variants, or choosing treatments based on tumor genetics, will be standard practice globally, like how antibiotic selection is standard today. Intermediate personalization, regular proteomics screening, and AI-assisted diagnosis will be available in most developed healthcare systems and expanding in middle-income countries. Advanced personalization, sophisticated digital twins, and real-time biological monitoring with continuous optimization will be accessible to larger populations as costs decline and technology matures. The patient experience will transform fundamentally. Healthcare will shift from episodic clinic visits when something goes wrong to continuous partnership between patients, physicians, and AI systems monitoring health constantly and intervening precisely when needed. Medicine will feel less like crisis response and more like cultivation, tending to your health continuously to keep all systems functioning optimally. Physicians' roles will evolve significantly. They'll spend less time on routine diagnosis and treatment selection, tasks where AI excels, and more time on complex decision-making, patient communication, and managing the human dimensions of healthcare that no algorithm can replace. The doctor-patient relationship remains central, but it's augmented by computational tools that make every physician's expertise more powerful and precise. This is the promise of personalized medicine built on biotechnology, a future where treatment is tailored to individual biology, where diseases are predicted and prevented rather than merely treated after symptoms appear, where every therapeutic intervention is optimized for maximum benefit and minimum harm, and where health is monitored and maintained continuously rather than repaired episodically. By 2050, the idea of prescribing the same medication at the same dose to millions of different people will seem as outdated as bloodletting seems today. We'll wonder how medicine ever functioned without understanding each patient's unique biological reality. There's a waiting list that haunts healthcare systems worldwide. Over 100,000 people in the United States alone are waiting for organ transplants at any given time. Kidneys, hearts, livers, lungs, pancreases. Some will wait years. Many will die before an organ becomes available. Even those fortunate enough to receive transplants face lifelong immunosuppression to prevent rejection, with all its complications and risks. This system, dependent on tragedy to provide organs and rationing a desperately scarce resource, represents one of medicine's most painful limitations. That limitation is ending. Not tomorrow, but systematically, through regenerative medicine technologies that will transform organ failure from a crisis of scarcity to a solvable engineering problem. By mid-century, the idea of waiting years for a donor organ will seem as archaic as waiting for scarce antibiotics once did. Regenerative medicine rests on a fundamental insight. The human body already knows how to build organs. Every person started as a single cell that divided, differentiated, and organized itself into a complete human being with dozens of organ systems. The genetic instructions for building hearts, kidneys, and livers are present in every cell. The challenge isn't inventing new biological processes, it's recreating and controlling the processes that built our bodies in the first place. The simplest form of regenerative medicine is something the body does naturally: healing. When skin is cut, it repairs itself. When bone breaks, it knits back together. Liver tissue can regenerate substantially after injury, but most organs have limited regenerative capacity. Hearts don't repair significant damage from heart attacks. Spinal cords don't reconnect after injury. Pancreatic beta cells, once destroyed in type 1 diabetes, don't regenerate. Extending and enhancing the body's natural regenerative abilities is regenerative medicine's first frontier. Stem cells are the key to regeneration. These are cells that can both divide to make more of themselves and differentiate into specialized cell types. Embryonic stem cells can become any cell type in the body. Adult stem cells, found in various tissues, have more limited potential, but can still generate multiple cell types within their tissue of origin. The discovery of induced pluripotent stem cells in 2006 by Shinya Yamaguchi revolutionized the field. Scientists learned to reprogram ordinary adult cells back to an embryonic-like state, creating cells with unlimited potential without needing embryos. This breakthrough solved both an ethical problem and a practical one. Embryonic stem cell research raised ethical concerns about embryo destruction. Induced pluripotent stem cells avoid this entirely, created from adult skin cells or blood cells. Practically, these cells can be made from a patient's own cells, creating perfect genetic matches that won't be rejected by the immune system. By 2025, stem cell therapies are already treating certain conditions. Bone marrow transplants, used for decades in cancer treatment, are essentially stem cell therapy, replacing a patient's blood-forming stem cells with healthy ones. Stem cell treatments for certain eye conditions are restoring vision. Engineered skin grafts grown from stem cells are treating severe burns. Cartilage regeneration using stem cells is being tested for joint injuries. But these are early applications, relatively simple tissues. The holy grail is whole organs: hearts, kidneys, livers. Three main approaches are converging toward this goal, each at different stages of development. The first approach is growing organs from the patient's own cells using biological scaffolds. Take a donor organ, perhaps from a pig or human cadaver, and strip away all the cells using detergents, leaving only the extracellular matrix, the structural scaffold that gives the organ its shape and provides biochemical signals. Then seed this scaffold with the patient's own cells, allowing them to repopulate the structure. The cells follow the scaffold's architecture and chemical cues, organizing into functional tissue. This approach has succeeded for simpler structures. In 2006, a patient received a laboratory-grown bladder created using this technique, the first bioengineered organ transplant. Tracheas, blood vessels, and urethras have been successfully engineered and implanted. Complex organs like hearts and kidneys remain challenging because they require multiple cell types precisely organized with intricate blood vessel networks, but progress continues steadily. The second approach is three-dimensional bioprinting. Imagine an inkjet printer, but instead of ink, it deposits living cells suspended in gel-like bioink. Layer by layer, the printer builds up three-dimensional tissue structures following a digital blueprint. The cells then mature and organize, forming functional tissue. Bioprinting sounds like science fiction, but it's already creating functional tissues. Research groups have printed skin for burn victims, cartilage for joint repair, and vascularized tissue patches. In 2019, researchers at Tel Aviv University printed a small heart complete with blood vessels, chambers, and tissues using a patient's own cells. It wasn't functional enough for transplantation, but it proved the concept. The challenge with bioprinting is resolution and vascularization. Organs need blood vessels reaching every cell. Networks of vessels at multiple scales, from large arteries down to tiny capillaries. Printing these intricate networks with the necessary precision remains difficult, but is improving rapidly. New bioprinting technologies can create vascular networks within printed tissues. New bioinks support cell survival and organization better. By the 2030s, bioprinted organs will likely move from research labs to initial clinical trials for simpler organs, then gradually advance to more complex ones. The third approach, and perhaps the most transformative long-term, is growing human organs inside animals. This sounds provocative, but the concept is scientifically straightforward. Scientists create an animal embryo, typically a pig or sheep, genetically modified so it cannot develop a particular organ, say a pancreas. Then they inject human pluripotent stem cells into that embryo. As the animal develops, the human cells preferentially fill the organ niche that the animal's own cells cannot fill. The result is an animal with a human organ growing inside it. This chimeric approach raises obvious ethical questions, which are being debated seriously, but it also offers enormous potential. Pigs are an ideal size for growing human-sized organs. Their gestation period is short, and crucially, the organ develops naturally, not assembled in a laboratory, resulting in fully functional architecture with complete vascular networks. Research in this area has progressed carefully under strict ethical guidelines. Scientists have successfully grown rat pancreases in mice and mouse pancreases in rats, then transplanted them to cure diabetes in recipient animals. In 2019, Japanese researchers received approval to grow human cells in animal embryos to study organ development, though with strict limitations preventing the embryos from developing to term. By the 2030s, assuming ethical frameworks permit, this technology could begin producing human organs for transplantation. A patient needing a kidney could have their cells reprogrammed to pluripotent stem cells, injected into a pig embryo modified to not develop kidneys, and 9 months later, a human kidney perfectly matched to the patient's genetics would be available. No waiting list, no rejection risk, no immunosuppression. Beyond whole organs, regenerative medicine is developing therapies to repair damaged tissues in place. If you have a heart attack, part of your heart muscle dies, replaced by scar tissue that doesn't contract. What if we could inject stem cells that become new heart muscle, restoring function? Multiple clinical trials are testing cardiac stem cell therapies with mixed but improving results. The challenge is getting injected cells to survive, integrate, and function properly in damaged tissue. Similar approaches are being developed for spinal cord injury, neurodegenerative diseases, and damaged joints. Stem cells might not replace entire organs, but could repair specific damage, preventing or delaying organ failure. A person with early kidney disease might receive stem cell therapy that regenerates damaged kidney tissue, avoiding the progression to complete kidney failure. Gene editing enhances all these regenerative approaches. If a patient's genetic disease causes organ failure, organs grown from their cells would have the same genetic problem. But if those cells are gene-edited to correct the mutation before growing the organ, the new organ would be healthy. A person with polycystic kidney disease could have their stem cells edited to remove the mutation. Then those edited cells used to grow healthy replacement kidneys. The timeline for these technologies reaching widespread clinical use varies by complexity. Simple tissues like skin, cartilage, and blood vessels are already in clinical use or late-stage trials. They'll be routine by 2030. Bladders, tracheas, and similarly structured organs will likely be available in specialized centers by the early 2030s. More complex vascular organs, like kidneys and livers, will probably reach clinical trials in the 2030s and potentially standard practice by the 2040s. Hearts, lungs, and other highly complex organs may not be routinely manufactured until the 2040s or later. But even partial progress transforms outcomes. If we can manufacture kidneys reliably by 2040, dialysis becomes a temporary measure rather than a permanent condition. If we can print skin unlimited quantities, severe burns become far more survivable. If we can grow liver tissue, acute liver failure becomes treatable. Each organ system conquered eliminates one form of previously fatal organ failure. The economic implications are substantial. The current transplant system is expensive. Organ procurement, surgery, lifelong immunosuppression, managing rejection episodes, and treating infection complications. Regenerative medicine will have significant upfront costs, but if it eliminates the need for immunosuppression and provides organs that function for decades without complications, the lifetime costs could be lower. Insurance models and healthcare systems will need to adapt. Current transplant waiting lists are managed through rationing based on medical urgency and compatibility. When organs can be manufactured on demand, allocation becomes less about rationing, scarce resources, and more about medical appropriateness and cost-effectiveness. Someone with end-stage kidney disease would receive a bioengineered kidney, not because they rose to the top of a waiting list, but because it's the medically appropriate treatment, just as someone with a bacterial infection receives antibiotics. Manufacturing capacity will be the new bottleneck. Specialized facilities will be needed to grow or print organs under sterile conditions with rigorous quality control. Initially, only major medical centers will have these capabilities. But like any manufacturing technology, capacity will expand as demand grows and techniques standardize. By 2050, organ manufacturing facilities might exist in most major cities in developed countries, with expanding capacity in middle-income nations. The psychological impact of regenerative medicine shouldn't be underestimated. Today, organ failure carries existential dread. The heart you were born with is failing, and there's no guarantee another will be available. This terror drives some people to desperation, seeking organs through ethically questionable means. In a world where organs can be manufactured, organ failure becomes serious but solvable, more like a mechanical problem than an existential crisis. Perhaps most profound is what regenerative medicine reveals about the nature of life and biology. For most of human history, the body was a given, fixed at birth, aside from growth and aging. Illness and injury were fates to be endured. Medicine could only support the body's own healing or manage symptoms. Regenerative medicine shifts this fundamentally. The body becomes something we can repair, rebuild, and even improve. Biology transitions from fate to engineering challenge. By 2050, regenerative medicine will have transformed organ failure from a death sentence or chronic condition into a treatable problem with manufactured solutions. The waiting lists will be historical footnotes. The question won't be whether an organ is available, but which method of regeneration is most appropriate for each patient. And humanity will have taken another step toward mastery of our own biology, building what evolution provided once, but now with intention and precision. You are not alone in your own body. For every human cell you call your own, there's roughly one bacterial cell living on or inside you. Trillions of microorganisms, primarily bacteria, but also fungi, viruses, and archaea, inhabit your skin, mouth, and especially your gut. Collectively, they're called the microbiome, and they carry about 20 million genes compared to your 20,000 human genes. In a very real sense, you are an ecosystem, a walking collaboration between human and microbial life that has evolved together for millions of years. For most of medical history, we viewed bacteria primarily as pathogens, invaders to be destroyed with antibiotics. This view wasn't wrong. Certain bacteria do cause devastating diseases, but it was radically incomplete. The vast majority of bacteria in and on your body aren't enemies. They're partners, performing essential functions your human cells cannot do alone. And we're just beginning to understand how profoundly these microbial partners influence health, disease, and even behavior. The human gut microbiome contains roughly 38 trillion bacterial cells, predominantly in the large intestine. These bacteria form complex communities with hundreds of different species coexisting, competing, and cooperating. Each person's microbiome is unique, like a fingerprint, shaped by genetics, birth method, early environment, diet, medications, and countless other factors. This microbial ecosystem performs functions so essential that we couldn't survive without it. Start with digestion. Humans cannot digest many complex carbohydrates and fibers. We lack the enzymes to break down these molecules. But gut bacteria possess these enzymes, fermenting indigestible fibers into short-chain fatty acids like butyrate, which our intestinal cells use as fuel. Without gut bacteria performing this digestive work, we'd extract far less nutrition from food. Fiber wouldn't just pass through unused. We'd lose a significant energy source and compounds that regulate metabolism and inflammation. The microbiome produces vitamins we cannot synthesize ourselves, including vitamin K and certain B vitamins. It trains and regulates the immune system, teaching it to distinguish harmless molecules from genuine threats. Mice raised in completely sterile environments without any microbiome develop severely defective immune systems. The bacteria literally educate the immune system from birth, calibrating its responses. Perhaps most remarkably, the gut microbiome communicates with the brain through what's called the gut-brain axis. Gut bacteria produce neurotransmitters including serotonin, dopamine, and GABA. They influence the vagus nerve connecting the gut to the brain. They affect the production of hormones and inflammatory molecules that impact brain function. Changes in gut microbiome composition have been linked to anxiety, depression, autism spectrum conditions, and even neurodegenerative diseases. This connection seems almost mystical, bacteria in the gut affecting thoughts and emotions. But the mechanisms are becoming clear. About 90% of the body's serotonin is produced in the gut, much of it influenced by gut bacteria. When bacteria ferment fiber, they produce short-chain fatty acids that can cross into the bloodstream, reach the brain, and affect neurological function. Inflammatory molecules produced during microbiome imbalance can trigger neuroinflammation. The gut and brain are in constant biochemical conversation, and the microbiome is a major participant in that dialogue. Disruption of the microbiome, called dysbiosis, is implicated in an astonishing range of conditions. Inflammatory bowel diseases like Crohn's disease and ulcerative colitis involve abnormal immune responses to gut bacteria. Obesity and metabolic syndrome correlate with specific microbiome compositions. Type 2 diabetes, cardiovascular disease, autoimmune conditions, allergies, asthma, and certain cancers all show associations with microbiome alterations. Even response to cancer immunotherapy appears influenced by gut bacteria composition. The critical question is causation. Does microbiome disruption cause these diseases, or do the diseases alter the microbiome? In many cases, the relationship is bidirectional. Certain microbiome compositions increase disease risk, but disease also changes the microbiome. Teasing apart these relationships requires careful research, and that work is happening now across thousands of laboratories worldwide. The most dramatic proof that microbiome manipulation can treat disease comes from fecal microbiota transplantation. This procedure is exactly what it sounds like: transferring fecal matter containing gut bacteria from a healthy donor into a patient's intestine. The concept is ancient. Traditional Chinese medicine described similar practices, but modern scientific application began with treating Clostridioides difficile infections, severe diarrheal disease often triggered by antibiotic use that wipes out protective gut bacteria, allowing C. difficile to overgrow. For recurrent C. difficile infections, fecal transplants are remarkably effective, curing over 80% of cases where antibiotics fail. The healthy donor microbiome reestablishes normal bacterial communities that outcompete and suppress C. difficile. This success prompted research into fecal transplants for other conditions. Early trials show some promise for inflammatory bowel disease, though results are mixed. Research is exploring applications for metabolic disorders, autoimmune conditions, and even neurological diseases. But crude fecal transplants are just the beginning. The future of microbiome medicine is precision engineering of microbial communities. Rather than transferring entire communities blindly, we'll identify specific beneficial bacteria and deliver them targeted fashion. Rather than hoping donor microbiomes establish successfully, we'll design synthetic bacterial consortia optimized for specific therapeutic purposes. This work is already underway. Several companies are developing live biotherapeutic products, essentially pharmaceutical-grade beneficial bacteria for treating specific conditions. These aren't probiotics sold in health food stores, which typically contain a few well-studied strains in modest quantities. These are carefully selected bacterial strains or consortia, grown under controlled conditions, formulated for optimal delivery and colonization, and tested in rigorous clinical trials like any pharmaceutical. One approach targets metabolic disorders. Certain bacterial species are more efficient at extracting energy from food, potentially contributing to obesity. Other species produce metabolites that improve insulin sensitivity and glucose metabolism. By modulating which bacteria predominate in the gut, metabolic function might be optimized. Some clinical trials have shown that transplanting microbiomes from lean donors to people with metabolic syndrome can improve insulin sensitivity, though effects are often temporary without sustained dietary changes. Another approach focuses on inflammation. Some bacterial species and their metabolic products are anti-inflammatory, others pro-inflammatory. Inflammatory bowel disease patients tend to have microbiomes skewed toward pro-inflammatory species. Rebalancing toward anti-inflammatory bacteria might reduce intestinal inflammation. Certain bacterial strains that produce high levels of butyrate, a powerfully anti-inflammatory short-chain fatty acid, are being developed as therapies. The gut-brain axis opens therapeutic possibilities for neurological and psychiatric conditions. If gut bacteria influence brain function, could we treat depression or anxiety by engineering the microbiome? Early research is suggestive. Some studies show that probiotics containing specific bacterial strains can reduce anxiety and depression symptoms, though effects are generally modest. More sophisticated interventions using targeted bacterial consortia might prove more effective. Parkinson's disease shows intriguing microbiome connections. Many Parkinson's patients experience gastrointestinal symptoms years before motor symptoms appear. Some research suggests abnormal gut bacteria might produce compounds that trigger the protein misfolding characteristic of Parkinson's, with these abnormal proteins spreading from the gut to the brain via the vagus nerve. If confirmed, this suggests microbiome intervention might prevent or slow Parkinson's progression. Autism spectrum disorders also show microbiome associations. Many autistic children have gastrointestinal issues and altered gut microbiome composition. Small studies testing microbiome transfer therapy in autistic children have shown some improvement in both gastrointestinal and behavioral symptoms, though much larger controlled trials are needed to confirm these effects. Beyond treating disease, microbiome engineering might optimize health. Personalized nutrition based on microbiome composition is emerging as a practical application. People with different microbiomes respond differently to the same foods. One person might experience blood sugar spikes from eating bread, while another person doesn't, partly due to how their gut bacteria process starches. Companies like Day 2 and Zoe are developing personalized dietary recommendations based on individual microbiome analysis and continuous glucose monitoring. The technology for microbiome analysis has advanced dramatically. Early studies used relatively crude methods identifying only major bacterial groups. Current sequencing technologies can identify hundreds of bacterial species and even strain-level differences. Metagenomic sequencing reads not just bacterial identity, but also the genes they carry and their functional capabilities. Metabolomics reveals what compounds the bacteria are actually producing. This detailed characterization enables precision microbiome medicine. Artificial intelligence is helping make sense of microbiome complexity. With hundreds of bacterial species interacting with each other, the host, and diet, the number of possible states is astronomical. Machine learning algorithms can identify patterns in this complexity, predicting which microbiome compositions correlate with health or disease, and suggesting interventions to shift composition toward healthier states. Looking toward 2050, microbiome medicine will operate on multiple levels. For acute conditions like C. difficile infection, microbiome restoration will be standard treatment, more refined than current fecal transplants, but based on the same principle. For chronic inflammatory conditions, targeted bacterial therapies will reduce inflammation and modulate immune responses. For metabolic health, personalized probiotic formulations based on individual microbiome analysis will optimize metabolism and prevent metabolic disease. Preventive microbiome care will become routine. Newborns might receive beneficial bacterial consortia optimized for immune system development. Children could receive microbiome support during antibiotic treatment to prevent lasting disruption. Adults might take periodic microbiome tests with personalized recommendations for maintaining healthy bacterial communities. Elderly individuals could receive interventions to counter age-related microbiome changes that contribute to immune decline and inflammation. The integration with other biotechnologies will be powerful. Gene editing could create bacterial strains with enhanced therapeutic properties, producing higher levels of beneficial compounds or more effectively suppressing pathogens. Synthetic biology might design entirely novel bacterial strains optimized for specific therapeutic functions. Personalized medicine approaches would combine microbiome analysis with genomic, proteomic, and metabolic data to create comprehensive health optimization strategies. Diet will remain central to microbiome health. No amount of bacterial supplementation can overcome a diet that doesn't support beneficial bacteria. Different bacterial species thrive on different nutrients, so dietary diversity supports microbial diversity. Fiber-rich foods feed beneficial bacteria that produce anti-inflammatory metabolites. Fermented foods provide live bacteria and support microbial communities. Understanding these connections will make dietary recommendations more precise and personalized. The ethical and practical considerations are manageable. Unlike gene editing, which permanently alters human DNA, microbiome interventions are temporary and reversible. Bacteria can be added, removed, or replaced without permanent consequences. This makes experimentation and optimization safer. The main risks are infection from pathogenic bacteria or disruption of beneficial communities, both manageable with proper quality control and monitoring. By mid-century, the idea that we ignored the trillions of microorganisms intimately involved in our health will seem bizarre. The microbiome will be recognized as an organ system in its own right, as important as the heart or liver, requiring care and maintenance. Medicine will treat not just human cells, but the entire human ecosystem, the holobiont of human and microbial life functioning together. And health will be understood not as a property of human cells alone, but as an emergent property of the complex collaboration between human and microbial genomes working in harmony. Every medical revolution faces the same fundamental question: who benefits first, and how does access spread? The most sophisticated gene therapy means nothing to someone who cannot afford it or doesn't live near facilities that provide it. The most advanced longevity intervention is irrelevant if only a small elite can access it. Understanding the economics and social dynamics of biotechnology diffusion isn't pessimism, it's realism, acknowledging that scientific possibility and practical availability are different challenges requiring different solutions. Let's start with an uncomfortable truth. Every transformative medical technology begins expensive and available only to the wealthy or privileged. This isn't unique to biotechnology. Antibiotics, vaccines, cardiac surgery, cancer chemotherapy, all followed similar paths. When penicillin was first produced during World War II, supplies were so limited that patients' urine was collected and the antibiotic extracted and reused. Early polio vaccines were rationed carefully. The first heart transplant in 1967 was a medical marvel available to exactly one person. Today, antibiotics cost pennies. Vaccines reach billions. Cardiac surgery is routine, and cancer treatment is available in most countries. The pattern is consistent: initial scarcity and high cost, followed by expanding production, declining prices, and broadening access. The questions are how fast this happens, what drives the transition, and how to accelerate it ethically. Consider genome sequencing as a case study. The first human genome cost approximately $3 billion and took 13 years, completed in 2003. By 2008, the cost had dropped to around $1 million per genome. By 2014, it reached $1,000. In 2025, whole genome sequencing costs a few $100s and takes days. This dramatic cost reduction, roughly 10,000-fold in 2 decades, resulted from technological improvements, automation, competition among sequencing companies, and economies of scale as demand grew. Gene editing therapies are following a similar trajectory, though they started more recently. Current CRISPR treatments like Casgevy cost around $2 to $3 million per patient. This price reflects genuine costs, research and development expenses, specialized manufacturing facilities, individualized production for each patient, regulatory compliance, and profit margins for companies that invested billions in development. The price also reflects limited competition. Only 1 or 2 approved therapies exist for these rare conditions, and recovery of development costs from small patient populations. But the cost is already projected to decline. Manufacturing processes are being optimized and scaled up. As more gene therapies gain approval, facilities can serve multiple product lines, spreading infrastructure costs. Competition is emerging as multiple companies develop therapies for the same conditions. And critically, the individualized production that makes current therapies expensive is being replaced by more standardized approaches that can serve larger patient populations more efficiently. Analysts project that gene editing therapy costs could decline to hundreds of thousands of dollars within a decade, and potentially tens of thousands within 15 to 20 years. This remains expensive, but it becomes comparable to other high-cost medical interventions like certain cancer treatments or long-term dialysis, interventions that healthcare systems already manage and insurance already covers. Insurance and reimbursement frameworks are adapting. Initially, insurers balked at multi-million-dollar one-time therapies, but the economic case for coverage is compelling. Compare a $3 million gene therapy that permanently cures sickle cell disease to lifetime management costs of hundreds of thousands or millions for hospital stays, transfusions, pain medication, and lost productivity. The gene therapy is cost-effective despite the high upfront price. Insurers increasingly recognize this, and regulatory pressure is pushing coverage of proven therapies. Government health systems face similar calculations. The UK's National Health Service approved Casgevy after negotiating confidential pricing discounts. Other national systems are evaluating coverage frameworks. The principle emerging is that genuinely curative therapies warrant coverage even at high initial costs if they reduce lifetime healthcare expenditure and improve patient outcomes. For longevity interventions, the economics are more complex because the benefits are preventive and distributed over decades rather than immediately curative. If a senolytic drug costs $10,000 per year and reduces age-related disease risk, should insurance cover it? The individual might live longer and healthier, reducing late-life medical costs, but they'll also accumulate more lifetime healthcare expenses by living longer. The cost-benefit analysis isn't straightforward. Preventive longevity medicine will likely start as out-of-pocket spending by wealthy individuals and gradually move toward insurance coverage as evidence accumulates. If metformin or rapamycin analogs prove to significantly delay multiple age-related diseases, the case for coverage strengthens. Insurance companies might even incentivize these interventions if they reduce near-term disease costs, similar to how some insurers subsidize gym memberships or smoking cessation programs. The geographic disparities in access are significant. Cutting-edge biotechnology requires infrastructure, specialized laboratories, trained personnel, regulatory oversight, cold chain logistics for biological products, and healthcare systems sophisticated enough to integrate complex new therapies. This infrastructure exists comprehensively only in wealthy countries. Middle-income countries have pockets of advanced capability, usually in major urban centers. Low-income countries often lack the foundational infrastructure entirely. This creates a biotechnology access gradient. Gene therapies, personalized medicine, regenerative treatments, and longevity interventions will be available first in North America, Western Europe, Japan, South Korea, and similar advanced economies. They'll expand to urban centers in China, India, Brazil, and other large middle-income countries. Eventually, as technologies mature and costs decline, they'll reach smaller middle-income countries and finally low-income countries. But this gradient isn't uniform across technologies. Some biotechnologies are more accessible than others. mRNA vaccines demonstrated that sophisticated biotechnology can be distributed globally relatively quickly when there's political will and massive investment. Billions of people in low and middle-income countries received mRNA vaccines within 2 years of their development. This happened because vaccines can be manufactured centrally and distributed. They don't require individualized production or extensive local infrastructure to administer. Gene therapies requiring patient cells to be collected, shipped to specialized facilities, edited, and returned are far more complex logistically. These will remain concentrated in advanced healthcare systems longer. But gene therapies delivered via viral vectors that can be manufactured in large batches and administered like vaccines could distribute more broadly, though still requiring trained healthcare workers and proper cold storage. Regenerative medicine faces similar logistical challenges. Growing personalized organs requires sophisticated facilities and cannot be distributed like pharmaceuticals. But if standardized bioengineered tissues become available, perhaps universal donor organs grown from genetically modified cells that evade immune rejection, distribution becomes more feasible. The role of international cooperation and policy cannot be overstated. The global response to COVID-19 showed both the potential and limitations of international collaboration. Vaccine development was remarkably rapid, a scientific triumph. But vaccine distribution was deeply inequitable. Wealthy countries secured supplies, while many low-income countries waited months or years. This pattern could repeat with biotechnology unless deliberate efforts ensure broader access. Several mechanisms could accelerate equitable access. Technology transfer programs, where companies share manufacturing knowledge with facilities in middle and low-income countries, similar to what happened with generic pharmaceuticals and antiretroviral drugs for HIV. Tiered pricing, where companies charge different prices in different markets based on ability to pay, allowing profit recovery in wealthy markets while providing affordable access elsewhere. Public investment in biotechnology development, with requirements for affordable access as a condition of funding. International pooling mechanisms, where countries collectively negotiate for access to technologies. The COVAX initiative, despite its limitations, demonstrated that international cooperation on health technology access is possible. Similar mechanisms could be developed for biotechnology, pooling purchasing power and negotiating access for participating countries. The infrastructure built for vaccine distribution could be leveraged for other biological therapies. Within countries, addressing disparities requires deliberate policy. Even in wealthy countries, access is uneven. People without insurance or with inadequate insurance may not access expensive therapies. Rural populations may lack nearby facilities offering advanced treatments. Minority populations often face systemic barriers to healthcare access. Making biotechnology available to all citizens, not just the wealthy or well-connected, requires strong public health systems, insurance mandates, subsidies for low-income populations, and investment in distributed healthcare infrastructure. The ethical dimension of biotechnology access is profound. If life-extending treatments are available only to the wealthy, inequality becomes literally embodied. The rich live healthy lives into their 90s, while the poor die in their 60s from treatable conditions. This scenario isn't hypothetical. It's already reality to some degree. Life expectancy in wealthy countries averages 10 to 15 years longer than in poor countries. Within wealthy countries, life expectancy varies significantly by income, education, and race. Biotechnology could widen these gaps or help close them, depending entirely on policy choices. Some argue that initial unequal access is acceptable if it leads to eventual broad access. The same argument made for smartphones, air travel, and other technologies that started exclusive and became common. Others argue health is fundamentally different, that allowing some to die from treatable conditions while others receive life-extending therapies is morally unacceptable regardless of future access expansion. Both perspectives have merit. Historical patterns do show that medical technologies eventually diffuse broadly, but eventually can mean decades, and lives lost during that time matter. The challenge is balancing incentives for innovation, which often require allowing companies to profit from new technologies with rapid access expansion that saves lives as quickly as possible. One promising model is advanced market commitments, where governments or international organizations commit to purchasing large quantities of a technology once developed, guaranteeing a market and allowing developers to recover costs while ensuring rapid distribution. This approach helped accelerate vaccine development and could be applied to other biotechnologies. Another approach is public funding for biotechnology development with strings attached. If taxpayer money funds research, perhaps the resulting therapies should be available at cost or with mandated affordable pricing. Several high-profile biotechnology breakthroughs, including CRISPR and mRNA vaccines, built on decades of publicly funded basic research. Ensuring public benefit from publicly funded science is both fair and pragmatic. Looking toward 2050, the most likely scenario is neither dystopian permanent inequality nor utopian universal access, but a messy middle path with progress and persistent gaps. Advanced biotechnologies will be routine in wealthy countries and increasingly available in middle-income countries. Low-income countries will lag but not be entirely excluded, especially for technologies that can be manufactured centrally and distributed broadly. Within countries, access will expand as costs decline and political pressure for equity grows. But disparities will persist based on insurance status, geography, and healthcare system quality. The degree of inequality in 2050 depends heavily on policy choices made in the 2020s and 2030s. Whether societies prioritize equity and invest in healthcare infrastructure and universal coverage. The biotechnology revolution has extraordinary potential to extend healthy life and reduce suffering. But potential means nothing without access. The technical challenges of developing gene therapies, longevity treatments, and regenerative medicine are being solved in laboratories worldwide. The economic and social challenges of ensuring these technologies benefit humanity broadly, not just a privileged few, require political will, international cooperation, and sustained commitment to health equity. These challenges are no less important than the scientific ones, and they will determine whether the biotechnology revolution fulfills its promise or simply adds another dimension to existing inequalities. Imagine being 75 years old in 2050. Your body carries genetic edits made decades earlier that corrected a predisposition to heart disease. Your microbiome has been carefully cultivated to support metabolic health and mental clarity. Twice a year, you receive senolytic treatments that clear accumulated senescent cells, keeping your tissues functioning like those of someone decades younger. Your digital twin, built from continuous health monitoring, predicted and prevented 3 potential health crises before symptoms ever appeared. Physically and mentally, you feel like someone in their 50s felt a generation ago, and you're looking forward to another 20 or 30 years of active, engaged life. This isn't science fiction, and it isn't utopia. It's a plausible scenario based on technologies already in development, but it raises questions that go far beyond biology and medicine. What does it mean to be human when we can edit our genetic code? How do we structure society when people routinely live healthy lives into their 90s and beyond? Where is the line between treating disease and enhancing human capabilities? And perhaps most fundamentally, what is the purpose of a longer life? Let's start with identity and what it means to modify our biology. For most of human history, our bodies were fixed givens. You were born with certain genetics, certain predispositions, certain limitations, and you lived within those constraints. Medicine could help you survive and manage problems, but it couldn't fundamentally alter your biological nature. That's changing. When someone receives gene therapy that permanently corrects a genetic error, they're altering the biological foundation of their existence. The question naturally arises, are they still the same person? Philosophy has long grappled with questions of personal identity. If you gradually replace every plank of a ship, is it still the same ship? If you alter your memories, are you still the same person? Gene editing adds a new dimension. Your genome is part of what makes you individually unique. Altering it changes something fundamental. but does it change who you are? Most people intuitively feel that treating disease doesn't change identity. Someone cured of sickle cell disease through gene editing is still themselves, just healthier. The genetic change removes an error, restoring function closer to typical human biology. This feels different from enhancement, deliberately improving capabilities beyond normal human range. The former is fixing what's broken. The latter is upgrading what works. But the boundary is fuzzy. Is preventing Alzheimer's in someone genetically predisposed treatment or enhancement? They don't have Alzheimer's yet, so technically, you're enhancing their future health beyond what their genetics would naturally provide. Is editing genes to improve immune function, treating immunodeficiency, or enhancing health? If we edit out genetic variants that increase diabetes risk, are we treating a condition or simply optimizing normal variation? These questions don't have universal answers. Different cultures and individuals will answer differently based on their values and philosophical frameworks. What's important is maintaining space for these diverse answers, not imposing a single view of acceptable biological modification. The enhancement question becomes more pressing as technologies advance. We know genes associated with intelligence, athletic ability, appearance, and personality traits. Technically, many could eventually be edited. Should we? Even asking this question makes many people uncomfortable, evoking memories of eugenics and coercive programs to improve the human race. Those horrors cannot be repeated, but there's a profound difference between government-mandated eugenics and individual choice about one's own or one's children's genetics. Liberal societies generally respect individual autonomy in medical decisions. People choose whether to take medications, undergo surgeries, or use reproductive technologies. If gene editing for enhancement becomes safe and available, restricting adult autonomous choice becomes difficult to justify philosophically, even if many find the practice troubling. The stronger ethical concern is children. Parents make medical decisions for children, but there are limits. We don't allow parents to refuse life-saving treatment based solely on preference. Genetic enhancement of embryos raises unique concerns because children can't consent. Effects are permanent and mistakes are irreversible. Most ethical frameworks support a cautious approach. Allow genetic modification only for clear disease prevention, not enhancement, at least until we understand long-term effects and social implications more thoroughly. Society will also face collective decisions about acceptable uses. Even if individuals have the right to pursue enhancement, societies can regulate how, when, and under what safeguards. We regulate pharmaceuticals, medical procedures, and reproductive technologies now. Gene editing for enhancement would likely face stringent regulation, requiring demonstrated safety, informed consent processes, and perhaps limits on what traits can be modified until we better understand implications. Then there's the question of inequality. If enhancement becomes available, initially, only the wealthy will access it. This could create biological stratification, where economic inequality becomes literally embodied in different human capabilities. The wealthy could edit their children's genes for intelligence, health, and longevity, while others cannot. Over generations, this could produce a genetic upper class and lower class, a dystopian scenario that troubles many thoughtful observers. This concern is valid, but it's not unique to genetic enhancement. The wealthy already provide their children with advantages: better nutrition, healthcare, education, social connections. These environmental advantages substantially affect outcomes. Genetic enhancement would add another dimension to existing inequality rather than creating inequality from scratch. The solution isn't necessarily banning enhancement, but ensuring broad access, so advantages don't concentrate in one group. This returns us to the equity questions from the previous episode. Policy choices will determine whether biotechnology exacerbates or reduces inequality. Beyond identity and enhancement, consider the social implications of significantly extended healthy lifespan. If most people live vigorously into their 90s, the entire life structure changes. Currently, people typically follow a pattern: education in youth, work in middle age, retirement in older age. This structure assumes declining capability after 65 or so. But if 75-year-olds have the capabilities of today's 50-year-olds, why should they retire? Extended working years could be liberating. Someone passionate about their career can continue decades longer. Someone wanting to change careers in their 60s or 70s has time to retrain and build a second or third career. The rigid age segregation of modern society, young people in schools, middle-aged people in workplaces, elderly people in retirement communities, could dissolve into more age-integrated communities where people of all ages work, learn, and contribute. But extended working years also raise concerns about stagnation. Do older workers block opportunities for younger ones? Do entrenched older people in positions of power prevent innovation and change? History suggests mixed outcomes. Some older individuals remain creative and open to new ideas throughout life. Others become rigid and conservative. Age alone doesn't determine this. Individual variation matters enormously. Mandatory retirement ages would likely become obsolete, replaced by capability-based assessments. If someone at 85 is mentally sharp and physically capable, there's no reason they can't work. If someone at 60 is declining cognitively, they might need to step back from demanding roles regardless of age. This shift from age-based to capability-based evaluation would be more fair individually, but more complex administratively. Retirement and pension systems would need fundamental restructuring. Current systems assume people work about 40 years and retire for 10 to 20 years. If people work 60 years and live in retirement for 30, the economics don't work. Solutions might include later retirement ages, higher retirement savings requirements, or new social models where people cycle between periods of work and leisure throughout life, rather than front-loading all leisure at the end. Family structures and relationships will evolve in less predictable ways. With extended lifespans, marriages could last 70 or 80 years. Will relationships endure that long, or will serial monogamy become more common? Might new relationship models emerge for extended life? Will people have children across wider age ranges, blurring generational boundaries? These questions have no certain answers, but they'll be worked out through lived experience as longevity increases. The psychological adaptation to longer lives is equally important. Purpose and meaning sustain people. If life extends, but purpose doesn't, additional years might feel empty rather than enriching. The challenge isn't just adding years to life, but adding life to years, ensuring the extended time is meaningful and fulfilling. Some worry that extended lifespans will lead to boredom or existential ennui. Wouldn't everything eventually feel repetitive? Human experience suggests otherwise. People find new interests, form new relationships, discover new purposes throughout life. Someone at 90 can be excited about learning a new skill or exploring a new place. The capacity for curiosity, growth, and meaning-making doesn't appear to have a fixed expiration date, though it does require cultivation. Others worry about overpopulation. If people live longer, won't Earth become overcrowded? This concern conflates two issues: lifespan and birth rates. Longer individual lifespans don't necessarily mean larger populations. Birth rates are declining globally, driven by education, women's empowerment, and economic development. Many developed countries already have below replacement fertility. Longer lifespans will age populations, creating more elderly people, but not necessarily more total people if birth rates remain low. The more pressing concern is resource consumption. Even with stable population numbers, people living longer consume resources for more years. But this must be balanced against reduced resource intensity from healthier aging. Healthy 90-year-olds consume far fewer medical resources than frail 70-year-olds requiring intensive care. Productive older people contribute economically rather than solely depending on younger generations. The net resource equation is complex. Environmental sustainability and biotechnology are not separate issues. The same scientific capabilities extending human life could help address environmental challenges. Synthetic biology might create organisms that capture carbon, break down pollutants, or produce materials sustainably. Biotechnology could reduce agriculture's environmental footprint by improving crop efficiency and reducing fertilizer needs. The biotech century could be more environmentally sustainable than the fossil fuel century, depending on how technologies are applied. There's also a question of gratitude and acceptance. Throughout history, humans have died from countless causes we now prevent or cure. Each generation has extended life and reduced suffering compared to previous ones. This is an extraordinary gift, the accumulated knowledge and effort of countless researchers, physicians, and thinkers who expanded the boundaries of what's possible. Extending this progress through biotechnology isn't defying nature or playing God. It's continuing humanity's long project of reducing suffering and expanding flourishing. But there remains the question of limits. Should humans pursue indefinite lifespan extension, trying to eliminate death entirely? Or is there wisdom in accepting mortality as part of the human condition? Different philosophical and religious traditions answer differently. Some emphasize acceptance of natural cycles and mortality. Others emphasize the value of life and the obligation to preserve and extend it when possible. Perhaps the question itself is premature. We're nowhere near eliminating death or achieving extreme lifespans. We're working on extending healthy years and compressing morbidity, goals that seem unambiguously positive. If we eventually face questions about radically extended lifespans or indefinite life extension, those generations will grapple with the philosophical and practical implications based on their experience and values. What's clear is that biotechnology is giving humanity unprecedented control over biology and health. This control brings responsibility. We must use these capabilities wisely, ensuring they reduce rather than increase suffering, that they're accessed broadly rather than hoarded by elites, and that they respect human dignity and autonomy. The biotechnology century will be defined not just by scientific capabilities, but by the choices we make about using them. The technologies emerging today will reshape human health, longevity, and perhaps human nature itself. Navigating this transformation requires both technical expertise and wisdom, both scientific rigor and ethical reflection, both bold innovation and careful consideration of consequences. By 2050, we'll be living in a world where genetic disease is increasingly rare and treatable, where aging has been partially slowed and its worst effects postponed, where personalized medicine tailors treatment to individual biology, where organs can be regenerated or manufactured, and where the microbiome is cultivated for health. This world will have its own challenges, questions we can't fully anticipate from 2025. But it will also be a world with less suffering from disease, longer healthy lives, and expanded human potential. The quiet revolution already underway is becoming the foundation of a transformed future. And we're all part of this transformation, not as passive observers, but as participants making choices individually and collectively about what kind of future we want to create. The biotechnology revolution isn't something happening to us. It's something we're building together. One discovery, one treatment, one patient, and one ethical choice at a time.